Good evening, James. I'm still in Portland because I still have school. So this morning I gave a presentation on B-cell lymphoma, which is a cancer of your lymphatic system. But it brought us some really cool and interesting things that I didn't know about some things in biology, and I want to tell you about them because they're really cool. So B-cells are the things that secrete antibodies. So antibodies are these little Y-shaped proteins which basically bind to an antigen, which is basically any sort of disease or strange thing that your body doesn't want to have inside of it at all, marks them for destruction, and it marks them to be destroyed or removed. The crazy thing with antibodies is there are more antibodies in your body than there are genes in your body total. Now, if you think about it, you'd have one antibody for every single antigen that could possibly ever occur, which is a lot of antibodies. The question is, how do we create millions or billions of different antibodies if we only have about 20,000 genes? The first really big way that we do this is this thing called VDJ recombination. So the way this works is antibodies have a variable region, and that's the region that actually binds to the antigen. Each antibody has its own specific variable region that is different than another antibody. The big way that this variable region becomes, so varied, is this thing called VDJ recombination. So basically, in your genome, there's this really long chain of genes, which are either V, D, or J. So ready? We're going to do some math, and it's going to be a little crazy. You have some proteins that come through and actually cut the DNA and actually relocate different V and D and J segments together to make different things. We have about 40 V sections, 25 D sections, and 6 J sections. And that's just for heavy chain. In the light chain, you don't have a D segment, but you still have the 40 V segments and 5 J segments. If we do the math here, that is 1.9 times 10 to the 8th possible combinations. They also go through this thing called somatic hypermutation, in which basically this random protein runs over the variable region and then just mutates things randomly. The big issue with this is it's actually mutating the DNA, and it's actually cutting the DNA. It's actually causing a double strand break in the DNA. So your DNA is here, so your DNA is here, and it's breaking, which is a problem sometimes because you have to put it back together, but sometimes you kind of put the wrong things back together. What happens there is that you have this thing called the IgH promoter. Now, the IgH promoter is what controls how many antibodies are made. The issue with this is when something that isn't an antibody gets put underneath this promoter, you get a lot of that thing. Sometimes one of the things that gets placed next to this IgH promoter is this protein called gene called BCL2, which basically stops cells from dying. Which is a problem because you're making 5 million B cells per day. You're making 5 million of these a day, and they're not going away. This is how tumors happen, and it's kind of crazy and not fun. I just don't think it's so crazy that our body has made a way to create so many different things that can combat all of the different things that can come into our body and cause us harm with still only having 20,000 genes, and possibly even less than that. I've also been writing down things that I want to talk to you about, which is an issue because when I write things down, my handwriting is terrible, and I actually can't read one of the things that I wrote down. Familiarity. I have no clue what I wanted to talk to you about with familiarity. That's what I taught people about today, which is really fun. All right, I'll see you next time.